All right, guys, the purpose of this video is to go into an introduction to immunity. And this isn't an incredibly in-depth, like full-on immunology, sort of coverage. This is basic overview for A&P. And the first idea here is we have a bunch of different cell types that are all classified as white blood cells, but they do very different things. And so we're going to come back to these as we get through the notes, but I think it'll be a helpful reference to circle back to. All right, so the first important thing is to differentiate between non-specific responses and what we call adaptive responses. Non-specific just means the response is the same every time. There's nothing unique about inflammation for any particular infection. So whether it's bacterial or viral or any specific type of bacteria or virus, the inflammation is all the same. And the goal here is, one, we want to significantly increase blood flow. So blood vessels are going to vasodilate. And part of that is bringing more nutrients and more white blood cells to the scene. So in this picture, what you're seeing is this is actually the outline of a capillary, right? Another one up here. And these white blood cells, these are likely neutrophils. They're actually physically squeezing themselves through the wall of the capillary. That process is called chemotaxis. That is white blood cells responding to chemical signals from the site of the inflammation that's basically luring them out to where the infection is so that they can start fighting it and addressing it. Right? Again, non-specific just because it doesn't really change how it happens from one infection to another. It's just a general sign that something's wrong and white blood cells need to address it. All right, another example of a non-specific response is fever, again, for the same reason. Fevers aren't really different depending on the infection. It's just a matter of how much did your temperature rise. And a fever is caused by something. Whatever that something is, we call a pyrogen. Pyro, like meaning fire. And the two different categories, exogenous and endogenous, just mean where did it come from? So an exogenous pyrogen is anything that came in from the outside of the body. These could be things that trigger an allergic reaction. Toxins from bacteria, like endotoxin, is something produced by gram-negative bacteria. And they stimulate a fever in an unregulated way. These are the really dangerous ones because your body isn't in control at that point. That's when fevers can skyrocket out of control. All right. The endogenous side of things means your white blood cells in your body produced signals on purpose to raise your temperature, right? And that idea that a fever is almost always purposeful. 99% of fevers are coming from these endogenous pyrogens. And the idea is bacteria especially have a harder time dividing when fever's high. So to sort of test that hypothesis, this is some clinical data. This research group looked at people that had a beta hemolytic strep infection, which is serious stuff. That's like strep in your bloodstream. And they compared people that survived the infection to people that didn't. Significant number didn't make it. And they scored it based on a handful of symptoms. Were they confused? Were they in pain? Were they conscious? Did they have a fever? And did they have difficulty breathing? And then they did an odds ratio. An odds ratio tells you whether something is a sign that you're better off or worse off. Right? So the interpretation is a 3.3 says if a patient comes in confused, they are 3.3 times more likely to die than someone who isn't confused. Right, same thing, being unconscious, 6 times worse off. Difficulty breathing, 4.4 times worse. Interestingly, these two, pain and fever, were actually good signs. Less than 1 means if you have pain, you're 50% less likely to die. If you have a fever, you're 80% less likely to die, right? 0.2 meaning 20% of the average. And all this is saying is, it's not that having pain and a fever is necessarily a good thing. It's saying if you have beta hemolytic strep, you should be in pain and have a fever, because if you don't, that's a good sign that your immune system isn't even responding and doing anything about the infection. So it's a good indication that the immune system is actually aware and at least trying to fight the infection instead of just giving up. Okay, so at this point, we have an infection. The tissue is inflamed. We maybe have a fever. These are all non-specific things. On the cellular side, your white blood cells need to start figuring out whether they're dealing with something dangerous or not. And the first step in all that is phagocytosis. And phagocytosis is the same concept as endocytosis. The difference is in endocytosis, the cell is actually interested in what the endocytosed material contains. Here, we're just trying to destroy it. And so the way this typically happens is one of these white blood cells, these are your major categories of phagocytic cells, they're going to ingest the bacterium or the virus based on binding to receptors on their cell surface. These pathogen-associated molecular patterns means molecules that are commonly found in viruses and bacteria but aren't found on humans. Right? And once you ingest the virus or the bacterium, the phagosome, which is really just a modified endosome that merges with a lysosome, has a bunch of enzymes and reactive oxygen species and acid that chop the bacteria or virus into bits. And those little bits, we call them antigens, right? Those are proteins that are then displayed on the cell surface. And this cell becomes an antigen-presenting cell, or an APC. This is going to be critical later on. These cells are going to go around showing off those antigens, asking other white blood cells like B cells and T cells if they recognize that antigen, and see if you're going to have an immune response or not. Okay, that antigen-presenting cell, which can be a lot of different cell types, but is often a resident macrophage, which just means a macrophage that has taken up residence in a certain organ like the lungs or the skin or the brain. And it's going to take that antigen and present it, right? That's where the antigen-presenting cell thing comes in. And so this macrophage phagocytoses your pathogen, right? Shows off the antigens on its MHC and starts basically shopping around the immune system, asking lymphocytes whether they recognize it or not. All right, so this T lymphocyte, which is most likely going to be a helper T cell, if it matches, what it's going to do is start activating B lymphocytes, right? And B lymphocytes are the cells that make antibodies. And so the B lymphocyte has these antibodies stuck on its cell surface. If the antigen binds the antibody and the helper T cell also releases chemical signals like cytokines that stimulate that B lymphocyte, they'll start dividing. And the B lymphocytes are going to divide into plasma cells that start secreting tons of antibody out into the blood. That's where they get their name, plasma cells, because they secrete into the plasma. And we expect those antibodies to coat our bacteria, virus, whatever it happens to be, so that phagocytic cells know that they should mount a strong immune response. Okay, so the phagocytic cell can sort of weakly respond at the beginning. Once these antibodies coat the pathogen, then it's a full, all-in immune response. All right, this entire process of B cells and T cells for that matter, but we're going to focus on B cells. Once a B cell is activated, and remember, activated means two things. It means that an antigen, right, a protein on the surface of a virus or bacterium or whatever's infecting you, stuck to its antibody, right? The B cell receptor in this case is an antibody. It's just that we sometimes call it a receptor when it's stuck in the cell surface. So if antigen binds the antibody and it gets stimulation from this helper T cell, then B cell starts dividing. Some of those B cells will become memory B cells that hang around for later in case you get the infection again. Some of them will become plasma cells that secrete antibodies, again, out into the plasma to go bind to the pathogen. All right, so antibodies. Antibodies are really just proteins. They're actually like very large complexes of 4 different proteins that are all linked together. And an antibody binds to a specific binding site on an antigen. Right? That very specific binding site we call an epitope. Right? So if this is our pathogen, say this is a bacterial cell or a virus, it doesn't really matter. Bacteria and viruses have all sorts of different molecules on their surface. Right? So I'll make them different colors to differentiate. And the idea is the whole thing, the bacterium, is the pathogen. Each of these individual surface molecules is one antigen. And then if we blow it up really close and say, like, this is a big view of the antigen, the epitope is the very, very specific binding site where the antibody sticks. One individual antigen can have many, many, many different epitopes. And the idea is It doesn't really matter exactly which of these antibodies you produce. What matters is that you produce some of them, right? And so if, one person might make that antibody and a different person might make that antibody, it doesn't really matter, right? As long as we're tagging something on the pathogen, this antibody is just assigned to phagocytic white blood cells that they're supposed to attack and destroy whatever's on the other end, right? All right. The next issue is, remember, every B cell in your body initially makes a different antibody than every other B cell. They're unique. And the only way that's possible is there's rearranging of DNA that happens when B cells are made. And so B cells come from your bone marrow. When those cells are produced, we randomly scramble the DNA that codes for the variable region of the antibody, right? This antigen binding site, this is the variable region. Variable meaning it's different in every B cell because every B cell binds to a unique antigen, right? All of this stuff down here, we call that the constant region because it's always the same. That's the part that your white blood cells recognize. And so for that variable region, every time a new B cell is produced from stem cells in your bone marrow, we scramble its DNA to make a random variable region. And the idea is you can't know in advance which pathogens you're going to run into, which antigens. And so you have to play it like a lottery. Make millions and millions of different types of antibodies, hope that you made some that actually work, so that when you get the infection, they'll recognize it, and then we start copying them, right? Clonal expansion. All right, so the details of this, we're not getting into the genetics, but you have all of these different segments within the DNA that codes for antibodies that get scrambled and rearranged almost like randomly pulling letters out of a hat, like a Scrabble board, and throwing them down, hoping you spell a word. It doesn't happen very often, but if you try millions of times, eventually you will. The total diversity, just for effect, I want you guys to see 10 to the 14th. It's like 100 trillion possible antibodies that your body could make. You can't make them all, but you don't have to make them all. You just have to hope you get a couple that work. All right, so you've made your antibodies, you're having your response. The other cool thing about antibodies is that they can improve over time. And the way that happens is through something called affinity maturation. Affinity means binding strength, right? How tightly does the antibody stick to its antigen? And maturation is just meaning it's getting better over time. And so the way this works is somatic hypermutation means that your cells intentionally mutate the gene that codes for the variable region of the antibody, mutated on purpose. Some of those mutations will be worse at binding the antigen, and some of them will be better. The cool part is the ones that bind better will naturally be stimulated to get copied because binding the antigen is a big part of what tells the B cell to divide. And so you effectively get natural selection within the B cell population in the sense that when you get lucky and you make an antibody that works better, that just means it's going to get copied more because it binds to the antigen more strongly. And so over time, not only do you get more antibodies, but the average affinity also tends to go up. All right, and the way that we can measure that is antibody titer. Right, so this graph is showing antibody titer over time. Titer is really a combination of two things. It's a combination of affinity and the actual concentration of antibodies. So think of titer as like affinity multiplied by concentration. You can have a higher affinity, higher titer through titer binding or through just having flat out more antibodies total. All right, so if you're exposed to an antigen for the first time, it's going to take the better part of a week, week and a half until you can really start producing antibodies. These IgM and IgG are just different subcategories of antibodies. IgMs get produced first, IgGs later, but they're a bit more long-lasting. And like right about in here is when you're most likely to be having your symptoms, the infection's raging, viruses, bacteria are copying themselves. Once you start getting your antibody response, hopefully the amount of pathogen goes down, you're in the clear. These IgG antibodies will hang around for several weeks to make sure that it doesn't come back. All right, secondary response, right, you get exposed again. But this time, instead of taking 10, 12 days to get a strong immune response, those IgG antibodies come back fast, right, more like 4 or 5 days now. that fights off the infection much faster. So the second time, you technically probably still had an infection, but you fight it off fast enough because you can make antibodies faster because you had increased memory cell counts that pushed your antibody titer up higher faster. And affinity maturation meant that they probably have a higher binding affinity too. And so in the end, we get a higher titer the second time around. All right, this is also how vaccines work in the sense that a good vaccine basically just mimics the primary response, right? So that you get that antibody production, memory cell production, without having to actually get sick because the vaccine is just showing your immune system what the real thing looks like. Right, and a vaccine can be made out of a lot of different things. Some of the best vaccines are made from live but weakened or attenuated cells and viruses, basically the real thing that's been partially killed with either heat or acid or genetically weakened. And if they're, I should have said if they're treated with acid or heat, they'd be considered dead. If they're still alive but genetically modified, they would be considered live attenuated. Either way, the point is, make it close enough to the real thing that your immune system recognizes the antigens, but not actually capable of causing an infection. Okay. You can also use pure protein antigens that are either harvested from cells or synthesized. There's also DNA, and now with the advent of COVID vaccine, mRNA-based vaccines, which basically deliver the instructions to the cell for how to make the antigen, and then you get the immune response, right? So like in the COVID vaccine, the mRNA that codes for COVID proteins, spike proteins, is the vaccine. Your cells make those proteins. They're displayed on the cell surface, and that activates B cells and T cells to mount an immune response when they see the real thing, hopefully. All right, as always, I hope this is helpful. Follow along in the playlist and subscribe so you see all the videos in order. It'll make more sense that way. And see you for the next one.